So how do you study how these venoms act? And one of the ways to do it is to purify the different components of the venom. And you need an assay for that. So at the beginning of our work, we were interested in the components of the venom that potentially could kill people because they cause paralysis. And so our assay was to use mice. And what we would do is we would put mice upside down on a wire screen and inject different fractions of the venom. And some fractions had no effect at all, in fact, most fractions, but a few fractions caused the mouse to start to be paralyzed. And if it's paralyzed, it can't clench its paws and hang on to the wire screen, and so it would fall. And so what we did was we just characterized and purified those components that made a mouse fall. And that way, we isolated, using this assay, the first two components of the venom, and these are their structures. And we were surprised to find that the active components of the venom are like very small proteins called peptides. And so the first peptide we isolated acts very much like cobra toxin or bungara toxin from snakes. And the second peptide we isolated acts very much like the deadly toxin that you have in pufferfish. And so when you get stung by one of these snails, these geography cones, it's equivalent to being bitten by a cobra and eating a lethal dose of pufferfish at the same time. And these are the chemicals that really cause that. They're small proteins with the sequences that you can see here. Then our whole research outlook changed because a young undergraduate, 18-year-old kid came into the lab and he said, you know, you guys have been injecting the mice directly in their body cavity. I really think it would be better if you inject mice directly into their central nervous system. And I wasn't so persuaded by this, but the great thing about being at a university is the students do what they want. They don't necessarily follow the professor's advice. And that really is the source of a lot of creativity of the research in universities. And so what this kid found was that if he injected directly into the brain of a mouse, that each component of the venom, and each peak here is one component of the venom, caused a different kind of behavior in the mice. So while there were some components that paralyzed mice, others made them jump and twist as they're jumping. Others made them uncoordinated. And this major peak that I'm showing you here, it put mice to sleep. And we were just amazed because the mice would go to sleep and 12 to 24 hours later, they'd be perfectly fine. But that was only true if the mice were under 3 weeks of age. If they were over 3 weeks of age, then instead of going to sleep, they would climb the sides of their cages, run from corner to corner. And then other peptides made mice drag their back legs, or run around in circles, or swing their heads back and forth, or kick on their back and scratch, or tremble, or scratch, or convulse. And so the remarkable thing was all this stuff was found in one venom. And so that tells us that the venoms of these snails were extremely complex with lots and lots of bioactive things. And so we've been trying to figure this out ever since. So why are there so many peptides? And it turns out that some peptides are used to capture prey, some peptides are used to defend themselves against predators, and some are to deter competitors. And so every species of cone snail has a set of biological interactions. And each species is different. So a different conus species would have different prey, different predators, and different competitors. In other words, each species has its own particular ecological niche. And so the venom components are essentially mutated as species evolve so that each is very fit for its own particular ecological niche. So the study of cone snails, and specifically this one, led to the isolation of one of these venom components that turned out to have a use that we never expected. Today, that venom component is an approved drug for very severe pain. And so when your pain is so severe that even morphine doesn't help, then the component of this cone snail venom can help you in a lot of cases. So this was really unexpected that by studying this very seemingly esoteric group of snails, we were able to end up with a drug for pain that now is used by people in very severe pain. Thank you very much.